very much, Ron, for that generous introduction. Now I don't have to go talk, so I'll just take questions. Okay, so and I'm disclosing support from Novartis on some of the trials I'll discuss, and a small biotech that we found at the University of Pennsylvania called Community. And I want to warn you all, when I say CAR, it means a chimeric antigen receptor, not combination antiretroviral therapy. So we have two that influence in two different fields here, cancer and HIV. Now, my community slide is this, really the message is the yin and yang of cancer and HIV, and they benefit each other, so in many direct ways. So, and I'll discuss the cancer HIV wheel, a virtuous cycle where the fields have benefited each other. The use of CAR T-cells in HIV and in cancer patients. The rationale for synthetically enhancing our immune system, and that really means gene transfer and genetic editing. And the status of the evolving cell and gene therapy ecosystem. And Mike Latterman, who's close to the front here, published and had a great sort of a discussion and debate about this issue about curative intents in HIV and this evolving journal pathogen and immunity, so I highly recommend that. So, you know, I was here, been privileged to be here in the early 80s during the explosion of bone marrow transplant. And that was really the first form of immunotherapy when I was here that was really thought to work through supralethal chemotherapy. And now we know mostly what an allogeneic bone marrow transplant did in that setting was really, it was the incoming T-cells delivering anti-leukemic effects. Now, in the 80s, tumor-infiltrating lymphocytes and donor leukocyte infusions were given both for melanoma and for hematologic malignancies. And that was the first real human cancer immunotherapy where it began to work. But then in the 90s, we had the emergence of HIV, and we learned a tremendous amount by the study of the immune system then in patients with chronic infection, both CMV and HIV. And cytotoxic T lymphocytes that were given in the first really informative trials, actually here in the Fred Hodgepodge, Stan Riddell, and Phil Greenberg, and showed that you could take lethal CMV infection in an immunosuppressed host and cure it with a single infusion of CTLs. In 1996, we began using lentiviral vectors in a human trial to treat HIV patients, and that was the first time that HIV itself, modified with VSVG, was given to patients with HIV, and we found good engraftment and anti-HIV effects in those studies. This was initially an anti-gag, anti-sense that we gave an envelope, but those were the first days where we could show T-cell going up in patients following about the therapy. And then we began in '97 using CAR T-cells in patients, initially HIV and then later in cancer. And then, you know, this really seminal experiment happened with Tim Brown, who has been here most of the day, when 2007 he had a bone marrow transplantation and led to this one singular example of a cured patient now a decade out. So I think now we have a lot of information on how we can make this happen routinely. And just a little bit about what the basic for the virologist and non-immunologist. You need CD4 cells. HIV has taught us that. Without CD4 cells, you rapidly get opportunistic infections. And studies by many, including Mike Bevan here in Seattle, show that when you get initial antigen response, you get an acute response, and then innate immunity, and then the T-cell adaptive immunity happens. And then if you get a secondary exposure, the cardinal effect of that is that now is much more rapid and robust than the first time around. So that's a recall response. To have that memory be maintained, you need CD4 cells, and they need to work and be functional. But this has been shown now in both in chronic infections, vaccines, as well as in cancer patients. And there are different mechanisms. What cancer does is inactivate cells and lead to a state that's called exhaustion now, which is a cell that doesn't work. It basically takes up oxygen but can no longer proliferate and kill its target. And in HIV, obviously, the virus is oftentimes cytopathic, and so it deletes the CD4 cells. So in both cases, you end up with ineffective CD4 cells, whether you have cancer or HIV, leading then to CD8 cells don't work well. And that's a central issue in immunopathology. Now, we were fortunate to have Tim Brown visit Penn in 2012. Bruce Levine, who was a postdoc in my lab, worked out the system that grew T-cells that we use for both CARs and cancer patients and HIV patients. And we began working with Sangamo in 2002, approximately, to look at could we knock out CCR5 in patients with HIV. Because we knew, by work in a number of labs, that CCR5 was really redundant and not required for a healthy immune system. So it's the entry factor for HIV and transmission, and yet you can live without it. And that probably happened during the bubonic plague episodes in Northern Europe, and it's selected for people who had a SNP in CCR5 that made it non-functional. So what we tried to do, rather than do the allotransplant that Tim Brown had to go through, is could we do this autologously and use the patient's own stem cells or T-cells. And we've been using T-cells at Penn, and Jim Riley's going to discuss the most recent results later this afternoon, and Paula Cannon and a group down in Los Angeles are attempting to do this now with CD34 stem cells so that it can be done without the necessity of a transplant. So can we do this more than once? So Paula Cannon said this one time, which is, you can't read it here, you only have to hear one talking dog to know that dogs can talk. And so at this point, we only have one talking dog. We need many more to make us feel more vibrant. But one talking dog is enough to convince me it can be done. So what about cancer immunotherapy? It is, has actually much longer and disappointing history than HIV. The first cancer therapy efforts were talked about in the 1860s by Paul Ehrlich, and then done with injections of heat-killed bacteria in the 1890s as a way to stimulate immunity in patients for cancer. It never got FDA approved, but it did have regressions in some patients. We now know that was through TLR activation of the innate immune system. And so many experiments didn't work, but finally in 2011, a T-cell-based therapy was FDA approved, and that was ipilimumab, or anti-CTLA-4, and that's been a really turning point. And in that year, in 2013, in Science, the cover magazine of this Science was CAR T-cells and ipilimumab. So checkpoint therapy, either anti-PD-1 or anti-CTLA-4 and CAR T-cells have led to a major change now in acceptance of the field of cancer immuno-oncology. This is what happened then in 2013 when Science magazine recognized cancer immunotherapy. And so that year, number 2 out of the 10 listed things was CRISPR technology. So gene editing, which is an amazing thing, and that's now coming into, I think, will be part of the general solution for HIV cure effects. And my daughter is 14 years old, and this is her favorite on this list, number 8, which is why we sleep. So lots of things happened in 2013. And this is now from a review recently published from Axel Hoos about the pipeline in cancer immuno-oncology, which is, it began with a dendritic cell vaccine, which actually had it here in Seattle, and then for prostate cancer, and then checkpoint therapies, many of them. There's now more than 700 trials open within cancer patients with different kinds of checkpoint therapies all across the world. I mean, it's impressive. There are not enough patients to test all the ideas. The CAR T-cells are expected to get FDA approval in the next several weeks. And last year, or in 2015, a modified herpes simplex virus called TVEC was FDA approved. And this is a virus that secretes GM-CSF, and it's injected directly into metastatic lesions in melanoma. It probably primes the immune system against mutated antigens in the tumor. So we're going to have many, many different kinds of biologics and cell-based therapies, and the real challenge is going to be, how do we work these out and get in combinations. So a CAR T-cell is shown here on how they actually work. So our T-cells are clonal. We heard about how HIV can infect some clones and others, and then come out and reemerge. Each T-cell has its own T-cell receptor, about 10,000 per T-cell, and they're all specific for one pathogen. And then a CAR then can be introduced with a number of ways. We use lentiviral vectors, and then to constitutively express a CAR. So that means it's permanently expressed on the surface of the T-cell. And this then is a synthetic molecule that now can redirect the cell to any desired protein, in this case CD19, which is on leukemia cells. And when the CAR contracts and contacts the tumor cell, it will kill it. And it can't tell the difference between a normal B cell and a malignant B cell, so it kills all cells that have, in this case, CD19 on them. And it's very potent. The CAR T cell, importantly, remains now, it's a bispecific cell. It keeps its day job, if you will, which is maybe anti-CMV through its natural T cell receptor, and then the ability to be retargeted to antibody-based approach of what the CAR is specific for. So we began the first trial using this second generation CAR with CD19 introduced with lentiviral vector in July 2010. And those patients had chronic lymphocytic leukemia. And then we began in 2013 treating patients, 2012 actually, with acute lymphoblastic leukemia, ALL. And basically every B-cell tumor that we treated and have been treated here with a group here at the Fred Hutch has responded. So it's been quite remarkable, multiple centers now, that CAR T-cells work in a number of chemotherapy refractory tumors. And the approach in general is to use the patient's own peripheral blood T-cells, and then using synthetic engineering with a lentiviral vector and introduce a CAR. We cultured initially for 10 days, and now this can be down as short as 3 days, this manufacturing time. It's called the vein-to-vein time, from when the cells are harvested to when they can be reinfused. We discovered cytokine release syndrome, and I'll discuss about that. It didn't happen in our preclinical experiments. And we had our own, the emergence of our own Tim Brown, who is Emily Whitehead. So that's the field of immuno-oncology. And so it's been the poster child. So this was all talked about in August 2011 when our first 3 patients were published, and it came out really to the public of what synthetic biology was in this New York Times article. And they described this, the first CLL patient that we treated in 4 steps, about using a modified HIV virus, taking the patient's cells out, inserting the CAR, and then how these modified cells could be the first living drug, because they persist in the patients now after 7 years since we treated them. And then they have persistent immunosurveillance, and involved in probably why relapse is so rare. So a number of centers are now doing CAR trials for both acute lymphoblastic leukemia as well as CLL and other B-cell tumors. There's both companies doing this as well as academic centers. Novartis is shown here. This is Judo trials and Kite. They all have had very high response rates in ALL, between 70 to 90% CR rates. The details are different on how they're constructed, the various CARs. We're just in the very beginning in that. But the field almost was stopped by cytokine release syndrome. And what happened when we treated Emily Whitehead is shown here. These are cytokines on a log scale. So we treated her on a Tuesday, and these are days listed as Tuesday, Wednesday, Thursday, Friday. On Friday, her cytokine values for IL-6 and interferon gamma were up 1,000-fold above baseline. And I was actually out here giving a talk and had written a letter that she was going to die. She was multi-organ failure. She was on a respirator. And I got back that night values that are shown here from our lab, showing that she had very high levels of these cytokines. We didn't know what was going on. She was multi-organ failure in the ICU. But what we had was something actionable. We had an anti-IL-6 antibody in the formulary. My daughter had juvenile rheumatoid arthritis. It had just been approved for JRA, the tocilizumab.
And so I called up the ICU at the Children's Hospital and said, why don't you treat Emily with anti-IL-6? And they said, you're a cowboy. And the patient said yes, the parents did, and it happened. And within an hour, she woke up and was fine. And see, she is, as I mentioned, is Artine Brown. And this is her on the front page of the New York Times. This is Emily at the opening of the Precision Medicine Initiative at the White House with the previous administration in January of 2016. And she sat actually next to Kareem Abdul-Jabbar. He's 7 feet 1. He has chronic leukemia and is on a BCR-ABL inhibitor in remission. And she has acute leukemia and she's been treated with CARs. And in the receiving line that day, her dad, Emily's dad, met with Obama and said, Obama said, can I do anything for you? And he said, yes, she needs an excuse to get out of school. It's a true story. Our current administration would write an excuse that Barack Obama did. So this is what happens molecularly now with next generation sequencing and B-cell tumors. You can identify the malignant clone with very precision and to very low levels. And this is what happened with Emily at the time of we treated her. So this is the day before a bone marrow biopsy and in the blood. Here's the bone marrow in the blood. 99% of her cells were 1 B cell.